into mini lecture 2D. This time we're going into the cell division, the way that the cell divides and multiplies, and it's called mitosis. 2D. The life cycle of a cell, mitosis, is in two sections. There is a time of the cell to grow, and then there's also this time for the cell to divide. Mitosis. So the cell life cycle has two major periods of cell growth or cell division. The function of that cell division is to produce more cells for growth and repair. They need to replace the worn out, torn, injured cells. So then all the new cells, who are named daughters, must have that same genetic information. That DNA molecule that forms the chromosomes must be identical. A cell divides in order to multiply. Old cells into two new cells. Mitotic division produces two daughter cells with the same number of chromosomes as that parent cell. Cell division. So the function of cell division is to produce more cells for growth and repair. All new cells, which are called daughters, must have the same genetic information. That DNA, the deoxyribonucleic acid, found in the nucleus of each cell. The DNA molecule that forms the chromosomes must be identical. DNA is composed of building blocks of protein, and it is in the shape of a ladder-like spiral staircase. Cells divide in order to multiply. Mitosis, the life cycle of a cell. Now, certain types of cells in the body divide almost continuously, all the time, mainly through wear and tear. For example, stem cells in our red bone marrow produce more than 2 million red blood cells every second. However, mature red blood cells do not divide. So before a cell divides to form two cells, the chromosomes are precisely duplicated, and the cell undergoes what's called mitosis. During mitosis, a complete set of chromosomes is distributed to each end of the parent cell, and that nuclear membrane forms around each set. After the parent cell divides, each new cell contains the identical number and the types of chromosome present in that parent cell. Now, when a fertilized egg divides, each of the new cells receives a complete copy of all the genetic information. During development, hundreds of divisions take place. Most cells in our body contain a complete duplicated set of the original chromosomes contributed by the sperm and the egg. Developing sperm and ova undergo a special process called meiosis, which halves their number of chromosomes. So mitosis is a bit different. So the life cycle of a cell includes 5 sections, short with IPMAT, IPMAT. There is an interphase, there is a prophase, metaphase, anaphase, and telophase. The cell starts with its interphase. The cell prepares for division and grows in size. The DNA of each chromosome replicates itself, is copied. Then it moves into the prophase, where the chromosomes appear. Each chromosome is made up of two strands called chromatids, and they are attached at what's called a centromere. These started off in the nucleus as chromatins, those squiggly wiggly wormy thing in the nucleus. The chromatins now have turned into chromosomes. Then we have some scaffolding happening in the cell. Some spindle fibers appear to give it strength and to be able to hold it together. But the nucleus has disappeared. So now we're into the metaphase. These chromosomes align across the equator of the spindle fibers. Then, moving into the anaphase, each centromere divides into two and moves to the opposite poles, pulling its own chromosomes behind it. Now there are twice as many chromosomes as before. And it moves into the telophase, that the new chromosomes start elongating. Two nuclear membranes appear, enclosing each set of chromosomes. So now we have the nucleus back again, two nucleuses with their chromosomes, which will become chromatins again in this telophase. So now within these new nucleuses, the DNA is in there, the DNA making the cells exactly the same as they were before. And this happens continuously right through our lives. A cell is in the interphase for its longest time before it decides to divide. So mitotic division is the division of old cells into two new cells, which then produces two daughter cells with the same number of chromosomes as the parent cell. And all the information in those cells must be the same as the parent cell. The DNA is carried along continuously. So here is the diagram as per your textbook, showing you in a little bit more detail the different phases of the cycle of the cell, going through the interphase, going through the prophase, the metaphase, the anaphase, the telophase. The cell divides, producing two daughter cells. Each cell is identical to the parent cell, except for size. In the picture there, you can see the nucleus with all its chromatin in there, and outside those centrioles, which help divide and move to the north or south poles. The mitotic spindle is there developing. Metaphase, those spindle fibers attach to the chromosomes and line up along the equator there. And then they get separated. The chromosomes separate, and one set of chromosomes moves towards each of the poles whether it's north or south pole. Then the cell begins to return to its interphase conditions, and the complete set of chromosomes is grouped at each pole. The nuclear envelope forms around each of those chromosomes, and the cell begins to divide. Now, in number 6, 2 identical cells. So, protein synthesis. DNA is the master blueprint of our protein synthesis. These proteins are our key substances for cell life. They are the building material for life. They are our building blocks. The ladder-like DNA molecules are the building blocks of protein and are joined during protein synthesis. Each gene has a specific arrangement of pairs. That DNA stays inside the nucleus and is like a master file. Its information is not useful unless it is decoded. And our decoder is our messenger, our RNA, our ribonucleic acid. Our RNA is the carrier of the message for the nucleus to those ribosomes. RNA takes amino acids, the breakdown of proteins, the amino acids, to the ribosomes in order required by the gene. So the site of protein synthesis in the cell is the ribosomes. So we've come to our conclusion. Our cells must grow and divide to continue their important work within our body. If our cells did not grow and divide and multiply, then we would not be able to grow and multiply. Cells do not last forever. They become worn, torn, injured, and die, and will be replaced. Cells go through mitosis to produce 2 identical cells to continue their tasks. Mitosis ensures that each and every cell contains all the genetic information precisely distributed to each new cell. A fertilized ova goes through meiosis, which halves the number of chromosomes because the chromosomes are coming from the sperm and the ova. But with protein synthesis, our DNA will continue to be replicated during each mitotic division, helping us to continue our species. I do hope that you've enjoyed this exploration through our cells. That is the end of Chapter 2, Part A, their cells, their makeup, and their functions. Chapter 2 continues with Part B next, moving into the tissues, their makeup and functions.